So what I'm going to do here now is talk about carbohydrate digestion. And as we know, the first stage in catabolism is digestion. And this is the breakdown of food into smaller molecules. So digestion will begin in the mouth, continues in the stomach, and concludes in the small intestine. This is just a brief overview of carbohydrate digestion. So our dietary carbohydrates, starch, glycogen, sucrose, lactose. Digestion starts in the mouth. So alpha amylase, which is an enzyme, this is found in saliva. This catalyzes the hydrolysis of the glycosidic bonds in the carbohydrates. Remember, enzymes act as catalysts in biological reactions. Starch and glycogen from plants are hydrolyzed to give smaller polysaccharides and the disaccharide maltose. Okay, so alpha amylase will continue to act on the polysaccharides in the stomach until the alpha amylase is inactivated by the stomach acid. Because remember, these enzymes are proteins, and they might have other non-protein parts associated with them. But remember, a protein will start to denature, okay, when we have an acidic environment. So this is what happens in the stomach. The alpha amylase will become inactivated by the stomach acid. Alpha amylase is secreted by the pancreas, and it enters the small intestine, where the conversion of polysaccharides to maltose continues. And there are other enzymes that hydrolyze maltose, sucrose, lactose to glucose, fructose, and galactose. The monosaccharides are then transported across the intestinal wall and into the bloodstream. So glucose enters the cell from the bloodstream, and when it does, it is immediately converted to glucose 6-phosphate. Glucose 6-phosphate formation is highly exergonic, so there's a release of energy, and this is important, it is non-reversible. Why is that? Well, glucose 6-phosphate obviously has a phosphate associated with it, so it turns out that phosphates cannot cross the cell membrane. Once the glucose is converted to glucose 6-phosphate, there are several pathways available. It just depends on the body's needs. So if energy is needed, glucose 6-phosphate undergoes glycolysis, and that'll convert it to pyruvate, and then pyruvate will be converted to acetyl coenzyme A, which will then enter the citric acid cycle. If there is glucose in excess, it'll be converted to other forms for storage. Glycogen by the way of the Glycogenesis pathway, or converted to fatty acids by the lipid metabolism pathway. We haven't talked about lipid metabolism yet. It can also enter what we call the pentose phosphate pathway, and this produces two major products, NADPH and ribose 5-phosphate. Ribose 5-phosphate is needed for the synthesis of DNA and RNA, which we'll talk about in the next module. So let's go ahead and take a look at glycolysis. And this is a series of 10 enzyme catalyzed reactions that break down each glucose molecule into 2 pyruvate molecules, and also yields 2 ATPs and 2 reduced coenzymes. This is pyruvate. So let's take a look at what happens in the glycolysis pathway. So steps 1 to 3, all right? It turns out that the phosphorylation of glucose requires energy, and it gets that energy from ATP. This is a highly exergonic and irreversible step. Again, phosphates cannot cross the cell membranes. Step 2, isomerization of glucose 6-phosphate to fructose 6-phosphate. So that's a rearrangement, and basically a 6-membered ring is converted to a 5-membered ring with this group here. Let's go ahead and take a look here. Steps 1 to 5 of glycolysis break one glucose molecule down into D-glyceraldehyde 3-phosphate fragments. Okay, so let's take a look here at step 1. Oh, before I go any further, we have, actually there's an investment of 2 ATP molecules that is required. Okay, so that's for steps 1 to 5. And then when we get to steps 6 to 10, steps 6 to 10 occur twice for each glucose that enters in at step 1. So steps 6 to 10 produce 2 pyruvates, 4 ATPs, and 2 reduced coenzymes. And this is per glucose molecule. Okay, so let's go back up here. And so, first what we have here is our glucose. And as we said, glucose is converted to glucose 6-phosphate. So this requires energy from ATP. All right. So the glucose undergoes the reaction with ATP in order to produce glucose 6-phosphate and, of course, ADP. And this is a catalyzed reaction. Don't worry, you don't have to know the enzymes. So in step 2, as I said, there's an isomerization of glucose 6-phosphate, and so here's our glucose 6-phosphate, and it undergoes an isomerization reaction to produce fructose 6-phosphate. And again, this reaction is also catalyzed by an enzyme, the mutase enzyme. Okay, so that's glucose 6-phosphate isomerase. That would be the enzyme that catalyzes this reaction. So for step 3, we have the fructose 6-phosphate, and again, it reacts with ATP. So it's 2 molecules of ATP that have been reacted here, okay, from steps 1 through 3. And this yields fructose 1,6 biphosphate. So notice the addition of the other phosphoryl group, if you will. The fructose 1,6 biphosphate has a 6-carbon chain, and this is cleaved into 2 3-carbon pieces. And this is also an enzyme catalyzed reaction. The 2 products are dihydroxyacetone phosphate and D-glyceraldehyde 3-phosphate. Okay, so these are both 3-carbon sugars, and the only product here that continues in the glycolysis pathway is the D-glyceraldehyde 3-phosphate, so this one here. In step 6, we have 2 reactions that occur. So the glyceraldehyde 3-phosphate, that's this molecule here, okay? This is oxidized. Notice the oxidizing agent here, the NAD+. So it's oxidized to a carboxylic acid, and then it is phosphorylated by another enzyme. So for this reaction in step 6, we need both the reduced coenzyme nicotinamide adenine dinucleotide and an inorganic phosphate. Okay, so both are required for this reaction here. Here we have the 1,3 biphosphoglycerate. Okay, so again, this was step 6 was an oxidation. Okay, step 7 again, we have, now in step 7, this produces energy. We have the conversion of ADP to ATP. So there's the phosphate group from the 1,3-biphosphoglycerate is transferred to ADP to form ATP. Okay, so we have one molecule of ATP here. So we end up with 3-phosphoglycerate. And then in step 8, a phosphate group is then transferred from carbon 3. Okay, here's our phosphate group. So it's transferred from carbon 3 to carbon 2. So we can see that here. We see that transfer. Okay, so right here. Here's the phosphate group on carbon 3, and here it's transferred to carbon 2. And this produces 2-phosphoglycerate. Okay. And then step 9, this involves water. So water is lost from the 2-phosphoglycerate, and this produces phosphoenol pyruvate. This is a dehydration reaction. We've seen dehydration reactions before. Dehydration reaction. Okay. So then finally in step 10, we have the phosphate group from phosphoenol pyruvate is transferred to ADP to produce ATP, okay, and we end up with pyruvate, okay. So that gives you an idea of what's going on in the glycolysis pathway. So I went through all of this already as I showed you the glycolysis pathway. And so this just tells us what happens at each step. Step 4 and 5, cleavage and isomerization. And what's important here is that steps 1 to 5, energy is invested in this part of the glycolysis. 2 ATPs are required. And then steps 6 to 10, energy is generated. In other words, ATP is formed. Okay, so we have 2 ATPs that were spent in steps 1 to 5 and replaced in steps 5 and 7. Okay, and in step 10, it makes sense, that is highly exergonic, and the transfer of the phosphate group to ADP is irreversible. So the energy released is accounted for by rearrangement of less stable enol pyruvate to the stable keto form. 2 ATPs produced in step 10 are the profits. So 2 ATPs were invested in the beginning, and then the 2 ATPs produced in step 10 are what we call the profits. So the important part here is the result of glycolysis. We end up with 2 pyruvate molecules. Okay, so glucose is converted to 2 pyruvates, production of 2 molecules of ATP, and we also have the production of 2 molecules of the reduced coenzyme. So what happens to pyruvate? Well, it's going to depend on metabolic conditions. In other words, are we under aerobic or anaerobic conditions? Remember, aerobic is the presence of oxygen. Anaerobic would be in the absence of oxygen. So under aerobic conditions, pyruvate will be converted to acetyl coenzyme A, and it goes right to the citric acid cycle. Under anaerobic conditions, without the oxygen, right? There's a lack of oxygen. Pyruvate is then reduced to lactate. So let's take a look here. If the electron transport chain slows down because of insufficient oxygen, remember, we need oxygen, okay, for that electron transport chain. So if it slows down because of insufficient oxygen, NADH concentration will increase. And that means the oxidizing agent, NAD+, is in short supply. We need this NAD+ for glycolysis to continue. If it's in short supply, glycolysis cannot continue. So the alternative way to reoxidize NADH is important because glycolysis, which is the only available source of the fresh ATP, must continue. So this occurs because of the conversion of pyruvate to lactate. So here we have pyruvate, and we have NADH serving as the reducing agent, and this is reoxidized to NAD+, and that becomes available then for glycolysis. So this here is lactate. The lactate formation serves only one purpose, and that's the production of NAD+. Once oxygen is available again, the lactate is reoxidized then to pyruvate, and then the pyruvate can be converted to acetyl coenzyme A and go to the citric acid cycle. So let's take a look here at the total energy output from oxidation of glucose. And this is the combined result of glycolysis, conversion of pyruvate to acetyl coenzyme A, conversion of 2 acetyl groups to 4 molecules of carbon dioxide in the citric acid cycle, the passage of reduced coenzymes from each of these pathways that go through the electron transport chain, and the production of ATP by oxidative phosphorylation. The sum of the net equations for each pathway that precedes oxidative phosphorylation is shown right here. So we know that each glucose yields 2 pyruvates and 2 acetyl coenzyme A's. And what we go ahead and multiply the net equations for pyruvate oxidation and the citric acid cycle by 2. Okay? For each molecule of glucose, there are 4 ATPs formed. The remainder of that ATP is produced by the electron transport chain and oxidative phosphorylation. So for each molecule of glucose, there's a total of 30 to 32 ATPs produced per molecule of glucose. So that's a lot of ATPs and therefore a lot of available energy. What we want to look at is metabolism in fasting and starvation. So, think about being on a really strict diet. Let's say you're in starvation mode, right? So these metabolic changes in the absence of food are going to begin, of course, with a decline in the blood glucose concentration. And when that happens, we see an increased release of glucose from glycogen. So all cells happen to contain glycogen, but most of the glycogen is stored in liver and muscle cells. So free glucose and glycogen represent less than 1% of our energy reserves, and it's used up in about 15 to 20 hours of normal activity. So during the first few days of starvation, protein will be used up at a rate as high as 75 grams a day. Remember, you're always told that when you're on a diet that it's important to exercise as well because you start to lose muscle, okay? So that's very important. What will happen is, when this protein is being used up, lipid catabolism, so the breakdown of lipids, is mobilized, and acetyl coenzyme A molecules are actually derived from the breakdown of lipids. And this acetyl coenzyme A will start to accumulate. So that acetyl coenzyme A needs to be removed. and it is removed by a new series of metabolic reactions that transfer the acetyl coenzyme A into what we call ketone bodies. So here are ketone bodies. We have 3-hydroxybutyrate, acetoacetate, and acetone. Okay, so let's go back up here for a minute. Anyway, let's say starvation continues, and what will happen is tissues in the brain can actually switch over to producing up to 50% of their ATP from breaking down the ketone bodies instead of glucose. By about day 40 of starvation, the metabolism will stabilize at the use of about 25 grams of protein and 180 grams of fat each day. So as long as there's water available, an average person can survive in that state for several months. People that have more fat can actually survive longer, which makes sense. So let's take a look at the regulation of glucose metabolism. So normal blood glucose concentrations range from about 65 to 110 milligrams per deciliter. And here are two terms that you should know: hypoglycemia and hyperglycemia. Hypoglycemia is when the blood glucose level or concentration is lower than 60 milligrams per deciliter. In this case, a person will feel weak, they might sweat, they get the chills, fast heartbeat. In very severe cases, it can cause confusion, convulsions, coma, and even death. My dad happens to be diabetic, and he watches his sugar intake and carb intake and all of that. But when he goes out to exercise, a lot of times he'll feel the weakness, he'll start sweating, he feels clammy and chilled, and he knows that he needs to eat some sugar quickly. So he usually carries around some little candies with him while he's out exercising. Hyperglycemia, on the other hand, is higher than normal blood glucose concentrations, greater than 110 milligrams per deciliter. Okay, so at about 140 milligrams per deciliter, you're diabetic, and when blood glucose concentrations gets to 180 milligrams per deciliter, the blood glucose will show up in the urine. So blood glucose is regulated by two hormones. Insulin, we already know about that. When blood glucose concentrations increase, insulin is released. The function is to decrease the blood glucose concentration. And how does it do this? It speeds up the passage of glucose into cells for energy production. So it speeds up the conversion of glucose to glucose 6-phosphate. It also decreases the blood glucose concentration by stimulating synthesis of glycogen, remember, the storage form of glucose, proteins, and lipids. The hormone glucagon is released when blood glucose concentrations drop. So what it does is stimulate the breakdown of glycogen in the liver, and of course when glycogen is broken down, glucose is released. Proteins and lipids are also broken down so that amino acids and glycerol can be converted to glucose in the liver. So I do want you to know about those two hormones. So this is a nice little representation of what's going on. Blood glucose concentrations rise, insulin is released from the pancreas, and this in turn causes the glucose to enter the cells faster. Breakdown of glucose by glycolysis speeds up. Glycogen synthesis increases in the liver and skeletal muscles, and synthesis of lipids and proteins increases. When the blood glucose concentration falls, glucagon is released from the pancreas. Glucose entry to the cells will slow down. Glycogen breakdown in the liver speeds up. So breakdown of lipids and proteins to raw materials for glucose synthesis then takes place. And we'll talk about gluconeogenesis a little bit later. Okay, so let's take a look at diabetes And diabetes is a condition that's due to either insufficient insulin or a failure of insulin to activate crossing of cell membranes by glucose. In other words, the receptors on the cell do not recognize the insulin. So there are two types. Type 1 diabetes, it's insulin dependent, and type 2 is adult onset diabetes. Symptoms can include excessive thirst, frequent urination, abnormally high glucose concentrations in the blood and in the urine, and the body can waste away even though the person is on a good diet. Actually, my dad, he eats pretty well, and yet he's very, very thin. He has type 2 diabetes. Basically what happens, the available glucose does not enter the cells where it is needed, and it spills into the urine. So symptoms result when available glucose does not enter the cells where it is needed. Okay, so here, let's take a look at the metabolic pathways of glucose. We have glycolysis, lysis, that's decomposition. So that's where glucose is converted to pyruvate. Gluconeogenesis. Genesis is creation. So this is the synthesis of glucose from amino acids, pyruvate, and other non-carbohydrates. Okay. Again, glycogenesis. Again, genesis is creation. And this is where glycogen is synthesized from glucose. That's the storage form of glucose. And then glycogenolysis, lysis again, that's the breakdown. So the breakdown of glycogen to glucose. And then there's a pentose phosphate pathway, and this is the conversion of glucose to 5-carbon sugar phosphates. So let's take a look at each one of these. We're not going to go into any detail, but I do want you to know about glycogen metabolism. Glycogenesis, this is the biochemical pathway for the synthesis of glycogen. Glycogenolysis, this is the biochemical pathway for the breakdown of glycogen to free glucose. Glycogen is the storage form of glucose. And if you remember from a previous chapter, it's a branch polymer of glucose. Glycogenesis, the synthesis of glycogen, this occurs when glucose concentrations are high, begins with glucose 6-phosphate, and glucose 6-phosphate is isomerized to glucose 1-phosphate. So what you have is a glucose residue which is attached to uridine diphosphate. We see, so the uridine diphosphate here, this is the glucose here. And what happens is the uridine diphosphate will transfer glucose to a growing glycogen chain. And this is an exergonic reaction, so energy is released. This occurs in the muscle cells if immediate energy is needed, and in the liver it occurs when blood glucose is low. So these are some important facts to remember about glycogenesis. Gluconeogenesis, this is the pathway for making glucose from non-carbohydrate molecules. Things like lactate, amino acids, and glycerol. Remember glycerol from the fatty acids. This is very important during fasting and starvation and also intense exercise. This is an endergonic process, so it requires energy and it is non-reversible. So the lactate that's absorbed from the blood is converted to pyruvate, which is the reactant for the first step of gluconeogenesis. Let's take a look at glucose production during exercise. This is also called the Cori cycle. Gluconeogenesis requires energy. We said it's endergonic. So what will happen is during exercise, when this pathway is shifted to the liver, okay, so this here would be the liver. Okay, this is represents the liver. This represents the muscle. So when the pathway is shifted to the liver, then what happens is it frees the muscle from the burden of having to produce even more energy. So the lactate is sent to the liver, okay, right here, and it's converted back to glucose in the liver. The glucose then can return through the bloodstream to the muscle, and there it can be stored as glycogen or used for energy production. And that's going to depend on the energy needs. The Cori cycle is most efficient after the completion of exercise. Anyway, this concludes the module. If you have any questions on any of this, please contact me, and everyone have a nice night.